diseases that can affect any part of the body. It is characterized by the rapid formation of abnormal cells, which can also invade other parts of the body, which is known as metastasis. And metastasis is a major cause of death from cancer. Cancer is also a leading cause of death worldwide. For example, in 2008, 7.6 million deaths occurred due to cancer, and this is 13% of all deaths, so that's a lot. And it has been predicted that the cancer of death worldwide will increase up to 11 million deaths by 2030. So it's important to do research on cancer. The main types of cancer are lung, stomach, colorectal, liver, and breast cancer. And on lung cancer, my colleague Evelyn will give a presentation later, and she will talk about a new tool to detect lung cancer. Now, about the key risk factors for the development of cancer are a high BMI, a low fruit and vegetable intake, a lack of physical activity, the use of tobacco and alcohol. And important is that actually more than 30% of all cancers could be avoided by by modifying or avoiding these key risk factors. Now, how does cancer arise? Well, it arises from one single cell, and it's a multi-stage process. Most often, it's a progression from a precancerous lesion to a malignant tumor. Mm-hmm. And actually, it's the result of the interaction of the genetic factors of a person and 3 external agents, such as the physical, the chemical, and the biological carcinogens. But most importantly, also aging is a fundamental factor for the development of cancer. So a little bit more about cancer and aging. The incidence of cancer increases dramatically with age. For example, when we look at the province of Limburg in Belgium, we see that 6 out of 7 new cancer patients are older than 50 years, and 3 out of 7 are older than 70 years. And now the reasons for this increased incidence with aging is actually a combination of three things. So first of all, the cellular repair mechanisms are less effective in older people. Older people are also longer exposed to carcinogenic events, and also age-associated changes occur to the immune system with aging. So as you can see, the field of the geriatric oncology is a very important field, but there are not a lot of studies performed in this field. So the study I'm working on is a study that focuses on this field. It stands for Kanker bij Limburgse Ouderenproject. It's financed by Interreg and has different partners. And the main focus is actually to investigate the impact of cancer, aging, and their interaction on the long-term well-being of older cancer patients. The CLIMOP study is an observational perspective cohort study, which means that a group of people with a common characteristic, for example here it is age and cancer, are followed over a longer period of time. And perspective means that the group you want to study is determined before the study is started. So in the CLIMOP study we focus on 3 groups. One is actually the study group, which are the older cancer patients, and then we have 2 control groups. So one for age, which are the younger cancer patients, and one for cancer, which are the older non-cancer patients. And cancer patients we include in our study are those with a new diagnosis of breast, prostate, lung, or gastrointestinal cancer. So the main aim of the CLIMOP study is to investigate the impact of cancer diagnosis and treatment on the well-being and also the quality of survival of older cancer patients, because not only the prolongation of survival is important, but also the prolongation of the active life expectancy, and also to increase the quality of survival. But, okay, I went too fast. So important is that the impact of cancer diagnosis and treatment is highly dependent on the age of the patient. However, it is not always easy to assess the correct age of a patient, because there is an inter-individual variability between different patients of the same age. For example, when we look at 2 women of the same age, you will see that one is able to do a lot. For example, she does her groceries herself, while the other has someone doing it for her. So even if they have the same chronologic age, apparently they do not have the same physiological age. So the chronologic age is a relative incorrect measure for the functional and the health status of an individual. So there is need for measures for the physiological age. And in the CLIMOP study, we want to do this by determining well-being of the patient and also searching for molecular biomarkers of aging. Well-being of the patient in our study is determined via a personal interview, which is based on the CGA. And CGA stands for Comprehensive Geriatric Assessment. It's actually a multidisciplinary evaluation of an older people, of the comorbidity, the psychological status, the nutritional status of an older person. So that's quite extensive. But I will not focus on this part during this presentation. I will mostly focus on the search for molecular biomarkers. So for this, we collect biological material, which are blood samples and buccal swabs. But first, I want to give a little background on these molecular biomarkers, because when the study started, we had to go think which biomarkers are interesting. And we came up with the immune parameters and also the telomere length, because they are both associated not only with aging, but also with cancer. And that is what we want to investigate in the CLIMOP study, so we want to look at older cancer patients. So why the immune parameters? Because it is already known that with aging, changes occur to the immune system, leading to a decreased immune competence, which is also known as immunosenescence. And why do we want to look at the telomere length? Well, studies already suggested the telomere length as a good measure or biomarker for aging, since it reflects the age of an organism on the cellular level. However, for the telomere length, there is still a lot of controversy, and therefore we want to go deeper into this. So both are associated with aging, but also with cancer. For example, the immune system, we all know that it is important for the elimination of cancer cells. And for the telomere length, is also known to be associated with the risk of cancer and the prognosis in cancer. So first, I will focus a little bit on the immune parameters. I will give a little bit background, and then on how we try to investigate this in our study. So about the immune system and aging, like I already said, the changes occur to the immune system with aging, which are known as immunosenescence. And this is characterized by a decline in cell-mediated immunity, so particularly in the T-cell function. And more specifically, this means a decrease in the naive T cells, leading to a decreased ability to resist new infections. An increase in the memory T cells, but a decrease in the diversity, leading to a decreased ability to respond to reinfection or to retain memory for cancer antigens on relapsing tumors. Also important during aging, a surface marker that is expressed on the T cells is decreased, so that is CD28, and this decrease is also a known biomarker for immunosenescence. And finally, also a T cell proliferation decreases and IL-2 production decreases. And IL-2 is also important for T cell activation. So you can see with aging, the T cell function decreases. Now, the causes for these changes in the immune system seen with aging are not well understood, but could be explained by thymic involution. This is a major age-associated change occurring to the thymus, leading to a decreased production of naive T cells. And secondly, also a very important potential cause of these changes is a chronic antigenic stimulation that occurs during life. For example, a viral source such as cytomegalovirus is known to be a very important cause in immune aging, leading to a low-grade inflammation. And even studies have indicated that the immune signatures rapidly change to a more elderly profile on CMV infection. So CMV is a very important factor also. So that is about the immune system and aging. Now about the immune system and cancer. Like I already said, the immune system is important in the elimination of cancer cells, and this is also evidenced by the immunosurveillance hypothesis. But importantly, the immune system is not only protective for the host, but can also have a tumor promoting function. So, or a tumor promoting action actually. And this is also seen in the cancer immunoediting process, which consists of 3 phases. So in the first phase, you have the elimination. So the adaptive and the innate immune system work together to detect transforming cells, so a developing tumor, and destroy it before it becomes clinically relevant. However, tumor cells that can survive this elimination phase, they are kept in a latent state of dormancy, in a functional state of dormancy actually, by the adaptive immune system. However, tumor cells will fight against the immune system and can then escape the immune system by changing their tumor antigen. The immune system gets deteriorated, and even the immune system can induce immunosuppression from the immune system. So here I gave a little overview, so when you look at home, it's easier to find it back. So that is about the immune system and cancer. So as a conclusion, we can say that certain aspects of immunosenescence can prevent an effective immune response against cancer and contribute to the overall increased susceptibility to cancer with aging. So now, that was a little bit of background. So now about the study. For measurement of these immune parameters, for investigating these immune parameters actually, we look at our 3 groups. We collect blood samples from these groups. And it is very important to collect these blood samples at baseline, so before treatment or before surgery, since surgery or treatment can interfere with the parameters we want to measure. So from the blood samples, serum and plasma are isolated and stored at the UV lim at -80 degrees Celsius until the new parameters can be measured. And also white blood cells are isolated and they are measured immediately. So now we have been thinking what immune parameters could we select to investigate, and we came up with a specific cytokine profiles and with the IRP, the immune risk profile. Why these 2? First, the cytokine profiles have already been found to be associated with immunosenescence, so this is the aging part. And it is also found that it can prevent an effective immune response against cancer, like I already said in the immunoediting process. And this is the cancer part. So this explains the role of these specific cytokine profiles in tumor development and also in the increased sensitivity to cancer with aging. Moreover, these cytokines have also been found to have a prognostic value, both in cancer and aging. So for example, the high IL-6, IL-10, IL-8, and TNF-alpha have been found to be independent predictors of a low survival chance in cancer. And high IL-6 and TNF-alpha have also been found to be linked to mortality and frailty in the older population. Now just something about frailty, because it maybe is not clear to everyone. Frailty actually means in Dutch kwetsbaarheid. It's collected in the frailty syndrome, and it's a collection of symptoms that occur due to aging-related loss and dysfunction of skeletal muscle and bone. And this puts the older person at an increased risk of adverse events such as death or disability. So this is just in short to clear this up. But about the specific cytokine profiles, so prognostic value in both cancer and aging has been found. But what about the predictive value on mortality and frailty in the older cancer population? It's still unknown, and this we will measure via an ELISA. So secondly, we also want to focus on the immune risk profile. Why? Because it's a cluster of parameters that can be used as biomarkers of immune aging. It emerged from a Swedish longitudinal Octonona study, which investigated age-associated changes in several immune parameters in a free-living population older than 85 years of age. And in this study, the IRP was found to predict 2, 4, and 6 years mortality. So it is an important factor in aging, and it is factors that are included in the IRP are the CMV. So as you remember, I previously already said that the CMV is very important in immune aging. And other factors included in the IRP are an inverted CD4-CD8 T-cell ratio. a low B cell count, a high CD8, CD8 plus, CD28 minus T cell count. So like, as a reminder, I said that the CD28 is an important factor also, and decreased T cell proliferation is included in the IRP. So now, the predictive value on mortality in the older population has been found in this study, but it's unknown if it's also predictive of mortality under any other circumstances, such as cancer. So in this study, we want to investigate whether the IRP also has a predictive value on mortality or frailty in the older cancer population. So we want to measure this. For the CMV, we will measure it via ELISA, and for the other parameters, we will use effects analysis. So the effects analysis were already performed because, like I said, the white blood cells are isolated from the blood plasma, and immediately effects analysis is performed. However, when I was analyzing the effects results, there were some pitfalls that led to the fact that I couldn't conclude much from my results. So, for example, here I stated these pitfalls. So, like I already said, it's very important to collect these blood samples before treatment or before surgery, so at baseline. And that is not always easy because lung cancer patients or gastrointestinal cancer patients are mostly treated before that surgery is performed. So it's not easy to collect these blood samples before treatment. So I had only 66 samples to analyze. Also, it is not easy to collect blood samples from male patients. And like I said, the lung and gastrointestinal cancer patients, it's not easy to collect them. However, I was able to detect something interesting in my results. So I found three groups in the CD4-CD8 ratio, and as you remember, it's included in the IRP. Actually, this is an important part because that is the part that is included in the IRP. And when we compare the younger cancer patient with the older cancer patients, we see that there is a slight trend towards an increased inversion of the CD4-CD8 ratio. And this is what we expect according to the IRP. And these are promising results, but as you can see, the younger cancer patients are more than the older cancer patients, so it's not quite significant, and these are not enough patients to say something significant from. But it's promising, and we hope in the future with more samples that we find the same thing, and that would be a good result. So as a conclusion, we can say that if these immune parameters, if we can find that these immune parameters are predictive for frailty or mortality in older cancer patients, and if there is a difference with younger cancer patients, the more vulnerable cancer patients can be screened and a more appropriate therapy can be selected. And finally, that was a lot about the immune parameters, but also the telomere length is a main focus of our study. Human studies have already found a significant relationship between the telomere length and age-related conditions, disease, mortality. However, like I already said, there is a lot of controversy regarding this relationship, and therefore we want to investigate this, but not only the relation with aging, but also with cancer. So that is what we will do in the CLIMOP study. And for this, we collect buccal swabs from our three groups. They are dried for 24 hours at room temperature and then stored at -20 degrees until DNA can be extracted and telomere length can be measured. This measurement of telomere length will occur via quantitative PCR. And currently I'm busy with optimizing a protocol to measure these telomere length. So if we can answer the question whether the telomere length is a good biomarker for aging and what the influence of cancer is, also, again, vulnerable cancer patients can be selected and a more appropriate therapy can be selected. So as a conclusion, the CLIMOP study is actually a study that focuses to screen for vulnerable cancer patients using biomarkers such as immune parameters and telomere length. Here you can also see the websites from our study, so you can look up information there. So thank you for your attention. Are there any questions? No questions?